Hi guys, my name is Kayla Bertholf. I am a junior biochem and molecular biology major at the College of Worcester, and my 2020 Apex Fellowship experience was with the Arnosti Laboratory in East Lansing, Michigan. Here's a little bit about my experience. So, as I mentioned before, this summer I worked remotely with the Arnosti Laboratory at Michigan State University. This was originally meant to be a in-person experience, but it became remote at the onset of the pandemic. I originally found this experience through networking with friends, and I ended up reaching out to Dr. Arnosti to ask if I could work with him this summer. The Arnosti Laboratory is part of Michigan State University. The focus of this lab is gene regulation in the context of development, so they have multiple projects going on with multiple different people, but the members that I worked with specifically over summer were Dr. Arnosti, who oversaw the whole project, and then Anna Maria and Daruba. Anna Maria is a PhD candidate student who works in the lab, and Daruba is a graduate student who was directly overseeing me this summer. So I had a research goal this summer, and that was to use a bioinformatic approach to analyze the conservation of the C-terminal binding protein that I will refer to as CTBP throughout this presentation. And then at the end of this, I would help to write a manuscript that describes our findings. So what even is the C-terminal binding protein, or CTBP? CTBP is a transcriptional co-repressor, meaning that it works with other molecules such as proteins or enzymes to bind to the DNA and control when and where and how it is expressed. There's both a long and short form of this protein, and we looked at both. However, we will end up focusing more on the long form in the future. As you can see in the picture here, this is the structure of CTBP, and you can see different sites. So there's a substrate binding domain and a NAD plus binding domain. So this is just where the different interactions occur within this molecule. Why study CTBP in flies? So flies have a lot of similar genes to humans. Actually, 76% of the disease-causing genes in flies are the same or similar enough to those in humans, as you can kind of see in the diagram on the bottom how different parts align with flies and humans. And CTBP and other transcriptional co-repressors in the first place have a pretty big involvement in human diseases and some pretty severe diseases such as heart disease, cancer, and things like that. So by studying how CTBP works and understanding its function, we might be able to understand how it functions in causing cancer or heart disease. So it's pretty relevant. Throughout this study, we looked at flies and then mosquitoes. So we started with the Drosophila melanogaster flies, as you can see in red in the top little part of this tree. And then we moved along to mosquito groups, such as the Aedes aegypti, Anopheles gambia, down at the bottom. And all of these species are a member of the Diptera, or the winged insect family. So what exactly did I do? I used both an evolutionary approach and a bioinformatic approach to look at the actual genes of the flies and mosquitoes and look for patterns. So I used software to scan for similarities and differences among them, and then I would highlight it and bring it up, as you can see in this picture at one of our lab meetings that we had weekly over summer. And this brings me to another point of what I did. A big thing was communication. So I would communicate between the other lab members and I, and we would discuss our current findings, what the next step was, what we were going to do. Communication was pretty difficult because it was a online experience. So we had to meet through Zoom, we had to communicate through email, sometimes text messages, but that was a pretty big part of my internship as well. So what did I find in my work this summer? I found that the different isoforms of CTBP that had been previously discovered may play a role in its function, and there's different conservation among them. And I also found that there is evidence of conservation of the long form across Diptera, so in both fly and mosquito. And there's also some evidence of conservation in certain sections between fly and mosquito. So based on this, future work can look towards the long form of this protein and more intense studying of the conservation there, as this may play an important role in how it's regulated and how it functions. One of the really cool things about my summer experience was that I got to help write a research paper on our findings that will actually be published after the entire experiment's done. And in particular, I got to help with the methods section and the results section. And I also got to make images that will be included in the paper, such as one down below is one of the ones that may be included. And it was really cool to learn the writing process for papers. Although I've worked on papers in classes, it's like a lot more intense to write an actual paper, and there's a lot that goes into the revision and reviewing process and rewriting and rewording and making sure that everything's accurate than I had originally thought. So it was a really cool part of my experience. At the start of this experience, I really hadn't looked into molecular genetics as a possible field of study. I had heard about it before, but I'd never intensely looked into it or studied it. And I found that it's actually really interesting. There is a lot of relevance in this field, not just with what my IS might be, but also in any career I can see. I gained a lot of useful tools that will help me in my career in general. A lot of the background research skills that I did not have before that I now do, which is really, really cool. And this experience opened my eyes to a completely new field of study that I had not been aware of. And if you're interested in working with molecular genetics, I'd say go for it. It's really great, but it does require a lot of patience because there's a lot of just sifting through genes and looking at the very minute details that you need to notice. So it takes a lot of patience, but it can be very rewarding. Yeah, so thank you guys so much for interacting with my PowerPoint and learning about my summer experience. I really do appreciate it. And if you have any more questions, here's some information on how to follow up with Apex and learn more about Apex fellowships and presentations. Thank you guys!